everyone. So yes, in our previous sessions of this chapter, we have completed all the types of tissues, both meristematic and permanent tissues, and we have started tissue systems, of which we have clearly dealt with the epidermal tissue system of root, stem and leaf. Now we are going to start with ground tissue system. Second one is ground tissue system. Fine. So, in ground tissue system, if you see, ground tissue system includes all the regions of the plant body except epidermis and vascular appendages. In that case, what are the tissues that are included under ground tissue system? We have after epidermis, the second region is cortex. Cortex region is divided into 3 layers. It includes hypodermis, note down parallelism, hypodermis, general cortex and endodermis. Fine. After endodermis, the region is pericycle, which includes what? After endodermis, stellar region comes into picture. Stellar region or stele, which includes stellar region or stele includes pericycle, medulla and vascular bundles. But of these 3, vascular bundles will come under vascular tissue system. So, here under ground tissue system, we will look into pericycle, medulla or pith, and medullary rays. Here you see parallelly for ground tissue system, we will be having a comparative analysis in between root, stem and leaf. Fine. So, first we look into root. Root, stem and leaf, of which under root we have both dicot and monocot. Dicot and monocot, dicot stem, monocot stem, dicot leaf, monocot leaf. Okay. So, look at here first in root, hypodermis will not be present. After epidermis, it is directly general cortex. So, there is no hypodermis in both dicot root and monocot root, whereas general cortex is parenchymatous. General cortex is parenchymatous in both dicot root and monocot root. Whereas endodermis, if you see, that is the last layer of cortical region, endodermis will have suberin depositions in its radial and tangential walls. Okay. So, endodermal cells will be like this endodermal cells will be like this. So, they are long elongated cells which are made up of parenchymatous cells only, but in their radial and tangential walls, they will be having suberin depositions which are called as Casparian stripes. Okay. So, these suberin depositions are called as what? Casparian stripes. Those endodermal cells which do not have any suberin depositions are called as passage cells. fine. These are passage cells, and these cells with suberin depositions are Casparian stripes. Okay. Next is pericycle, fine. Pericycle is again parenchymatous in the root. Fine. Medulla or pith is also parenchymatous. Medullary rays also the same, parenchymatous. Clear, right? So, both in the dicot root and monocot root, under ground tissue system or fundamental tissue system, what did we observe, ma? There is no hypodermal region. General cortex is purely parenchymatous, and endodermis will have parenchymatous cells. Okay, endodermis is made up of parenchymatous cells, elongated cells, but then their radial and tangential walls are having what? Casparian stripes. Okay, those endodermal cells which do not have any Casparian stripes or suberin depositions are called as what? Passage cells. And then pericycle again made up of what? Parenchymatous. Medulla or pith, parenchymatous. Medullary rays are also parenchymatous. Now you see dicot and monocot stem. Dicot and monocot stem. Hypodermis in dicot stem is collenchymatous. Hypodermis is collenchymatous. If you understand properly this table, then the comparative analysis between dicot and monocot will be easy for you. Okay. Cortex, the hypodermis in monocot is sclerenchymatous. Fine. In dicot stem, hypodermis is collenchymatous. In monocot stem, hypodermis is sclerenchymatous. General cortex in dicot and monocot is what? Parenchymatous. Fine. Endodermis in dicot stem will have a starch sheath. It has a starch sheath, whereas there is no endodermis in monocot stem. Okay. And pericycle will have semi-lunar patches which are made up of sclerenchymatous tissue. Okay. Semi-lunar patches of pericycle in dicot stem are made up of what? Sclerenchymatous tissue. Fine. And then medulla or pith is again parenchymatous in dicot stem. Medullary rays are also parenchymatous, whereas in monocot stem you do not have any endodermis, there is no pericycle, there is no medulla or pith. There is completely parenchymatous general cortex in the monocot stem. In that parenchymatous general cortex only, vascular bundles will originate as schizolysigenous in origin, which means few of the parenchymatous cells will degenerate, and in that lost degenerated cells, air cavities or spaces are created wherein vascular bundles will be arising. So, such way of arising vascular bundles is called as what? Schizolysigenous in origin. Next, in dicot leaf and monocot leaf, if you observe dicot leaf and monocot leaf, clearly you see, ma, hypodermis is absent. Okay, cortex region, hypodermis is absent in dicot and monocot leaf. General cortex is purely, or the ground tissue system in dicot and monocot leaf is made up of what? Mesophyll cells. Ground tissue system will have what? Mesophyll cells. This mesophyll cells in dicot leaf is differentiated into what? Palisade parenchyma and spongy parenchyma. Okay. And in monocot leaf, if you see, you just have what? Spongy parenchyma. Fine. That is the only ground tissue system composition in dicot and monocot leaf. Once if we quickly look into the entire ground tissue system or fundamental tissue system, what is it actually? All the regions of a plant body except epidermal region and vascular bundles is said to be ground tissue system. If we see ground tissue system in a root, both dicot and monocot root, there is no hypodermis. General cortex is parenchymatous. Endodermis is made up of parenchymatous elongated cells, but those cells with radial and tangential walls occupied with suberin depositions are called as what? Casparian stripes. This Casparian strips will prevent the apoplastic movement, and from endodermis we can see only symplastic movement of water transport. Okay. After endodermis, the next region in the stellar region is what? Pericycle. Pericycle is parenchymatous again in the root. Medulla or pith is parenchymatous. Medullary rays are also parenchymatous. Next, in dicot stem, here you will see more region wise differentiation, region wise tissue differentiation you can see more in the dicot stem only, and most of the times questions will be raised from dicot stem and monocot stem itself. Just listen carefully ma. First, from hypodermis, dicot stem hypodermis is what? Collenchymatous. Monocot stem hypodermis is sclerenchymatous. Next, general cortex in both of them is parenchymatous. In monocot stem you do not find any endodermis, pericycle, medulla or medullary rays. Whereas in dicot stem, endodermis will store food material and they will be having what? A starch sheath. Pericycle is having semi-lunar patches called as sclerenchyma. Medulla or pith is parenchymatous. Medullary rays are also parenchymatous. And then dicot and monocot leaf, if you see, hypodermis is absent. The only general cortex region or cortical region you see is mesophyll tissue. Okay, the ground tissue system or fundamental tissue system of a leaf is just having what? Mesophyll tissue. That mesophyll tissue is differentiated into palisade parenchyma and spongy parenchyma in dicot leaf, wherein palisade parenchyma is vertically arranged under the dorsal surface, that is adaxial surface, upper surface, into vertical rows with least intercellular spaces, whereas spongy mesophyll cells or spongy parenchymatous cells, mesophyll cells are again parenchymatous, they are having what? Large air spaces in between them. Clear, right? Any doubts anywhere in the complete ground tissue? No, right? Now we will move to vascular tissue system. Just give me a moment, ma. Yeah. So we will see vascular tissue system. Take down. Vascular tissue system purely includes what? Vascular bundles. Vascular bundles. Okay. So these vascular bundles include what, ma? Xylem and phloem. So yesterday when we are discussing about xylem and phloem, these are the complex tissues, we have clearly learnt all the 4 components of the xylem tissue and 4 components of phloem tissue that you see in advanced plants, angiosperms. What are the 4 components of xylem? Tracheids, vessels, xylem fibers and xylem parenchyma. What are the 4 components of phloem? Sieve tubes, companion cells, phloem fibers and phloem parenchyma. These are the 4 components of phloem tissue. So xylem movement, xylem will conduct what? Water and minerals from roots to the tip of the plant body, and the direction is unidirectional transport. Unidirectional transport. Whereas phloem will transport the food material mostly in the form of sucrose, okay, from source to sink. From source to sink. So source always will be a mature leaf, ma. Source will always be what? A mature leaf. Sink can be a root, sink can be a young leaf, sink can be a bud, okay, sink can be a fruit, young fruit, developing fruit. So these all can be the sinks, okay. Transport of food materials or nutrients from source to sink is the phloem transport. So source, and there are many sinks. So it is multi-directional. Unlike xylem transport, phloem transport is what, ma? Multi-directional. Take down. It is multi-directional. Fine, right? Yeah. Now see here, these vascular bundles based on the based on the presence or absence of phloem, there are of two types, and based on the arrangement of xylem and phloem in the stellar region, they are again are of two types. Okay. First we will see based on the presence or absence of the phloem. Based on presence or absence of cambium. Remember this thing, xylem will always be close to the pith region, okay. It is present in the inner side, and phloem is towards the peripheral side, that is towards the pericycle. So here if you observe due to the presence or absence of cambium, right. So if there is a cambium in between, cambium is present in between xylem and phloem, so this is called as what? Open form of vascular bundle, okay. Open form of vascular system, okay. If there is no cambium between xylem and phloem, this is called as closed form of vascular bundles. Open form of vascular bundles can be observed in dicotyledons, dicots. Closed form of vascular system you can observe in monocots. Fine. This will conduct secondary growth. Cambium here will conduct what na? Secondary growth. Clear? First type is what? Based on the presence or absence of cambium, we have two types. One is open form of vascular bundle, second is closed form of vascular bundles. Open form of vascular bundles observed in dicotyledons, which will promote secondary growth. Closed form do not conduct any secondary growth. Clear, right ma? Next, based on the arrangement of xylem and phloem in the stellar region, we again have two types, okay. Take down. Based on the arrangement of xylem and phloem in stellar region. Fine. So here if you see, first is what? Xylem and phloem. In root we will observe now. If you have to take a steel, the outermost region is what? Pericycle. In that we have xylem and phloem. So here xylem and phloem are at different radius, yes or no? Xylem and phloem are present at different radii. So these are called as radial type of vascular bundles, okay. If the tissue which is present in between xylem and phloem here is called as conjunctive tissue, okay, and from the pericycle only the cambium will arise over here, okay. If there is a cambium that is formed in between xylem and phloem, that cambium will be formed only in the dicot root. That cambium arises from where ma? Pericycle, important point, okay, just a second. So a wavy cambium can be observed in between xylem and phloem like this. What is this? Wavy cambium. Wavy vascular cambium that is formed in between xylem and phloem, where few of the conjunctive tissue cells get meristematic, and this wavy cambium arises from pericycle. Okay, cambium arises from pericycle. Fine. This wavy cambium is observed only in the dicots. As such, in dicot root, what type of vascular bundles you will observe ma? radial and open type, because cambium is present in between xylem and phloem, right? Radial and open type of vascular bundles. In monocot root, if you see, monocot root, radial and closed type of vascular bundles are observed. Radial and closed. Fine? Any doubt anywhere, ma? Yeah, next. Stem. So here if you see, in stem we have both dicot stem and monocot stem, right? First you observe basic vascular bundle arrangement in the stellar region of the stem. So here if you see, ma, okay. So here if you see, at the center you have pith, okay, and xylem close to the pith, fine, phloem towards the pericycle. So here if you observe, xylem and phloem are at the same radii. When xylem and phloem are arranged on the same radius, this pattern of vascular bundle arrangement is called as conjoint vascular bundles, okay. This is called as conjoint vascular bundles. And here if you see, phloem is arranged towards the periphery and xylem towards the center, okay, phloem towards the periphery or towards the pericycle, xylem towards the pith, and two vascular bundles are lateral to each other. Such arrangement is called as collateral, conjoint collateral, okay. And here there is a cambium between xylem and phloem. When there is a cambium between xylem and phloem, this is called as open type. Fine. So in dicot stem, what type of vascular bundles you will see, ma? Conjoint, collateral and open. In monocot stem, cambium will not be present in between xylem and phloem, so you will observe conjoint and closed type of vascular bundles. So here if you see, in dicot stem, you can observe conjoint and closed. In monocot stem, in dicot stem it is conjoint, collateral and open. In monocot stem it is what, ma? Conjoint and closed. Fine. In leaf, both dicot leaf and monocot leaf, you can see conjoint and closed type of vascular bundles. Take down dicot or monocot leaf, you have conjoint and closed vascular bundles. Clear? Any doubt anywhere, ma? No, right? Now, we will look into the comparative analysis between dicot monocot root, dicot monocot stem and dicot monocot leaf. Yeah, here we are in comparative analysis between dicot monocot, epidermal tissue system, ground or fundamental tissue system that we have discussed just now, and vascular tissue system. Then the comparative analysis between dicots and monocots, root, stem, leaf is quite easy for you. So here if you see, first is epidermal region. The epidermal region in both dicot and monocot, they are having what? Elongated cells. They are made up of parenchyma and they will have unicellular hair-like projections. This unicellular hair-like projections are what, ma? Root hairs. Okay. As such, epidermis here is also called as epiblema or piliferous layer or rhizodermis. Fine. Epiblema, piliferous layer or rhizodermis. Next to the epidermis, as we have discussed, there is no hypodermis as such, we just have what? General cortex. General cortex both in dicot and monocot root is made up of what? Parenchyma. Fine. After general cortex, it is what, ma? Endodermis. Endodermis is the last layer of the cortical region in the root system. This endodermis is parenchymatous elongated cells, it is single layered, okay, whereas it has suberin depositions in radial and tangential walls, which are called as what? Casparian stripes. Clear, right? These are called as Casparian stripes. And next in the stellar region, if you see, pericycle is again parenchymatous. Next you have vascular bundles. So the only difference between dicot and monocot root is there is no cambium in monocot root. Okay, here in dicot root, cambium is present. Okay, and pith is small or inconspicuous, and you can see number of xylem and phloem bundles are 2 to 6. So you can call it as diarch to hexarch condition. Fine. Cambium is present in between xylem and phloem, and pith is small, okay, and number of vascular bundles will be 2 to 6, so it is diarch to hexarch condition. In monocot root, the only difference in between dicot and monocot, you see, monocot root, there is no cambium. Fine, there is no cambium, and pith is large. You can observe more than 6 vascular bundles, where you can call it as polyarch condition. Okay, you can call it as what? Polyarch condition. These are the major differences between the dicot root and monocot root. Any doubts anywhere, ma? Clear, right? Complete dicot root and monocot root comparison. Fine. And here from the pericycle, if you see, pericycle of the dicot root, from pericycle, 2 things will arise. One is lateral roots will arise endogenously. Second point is wavy cambium is formed in between phloem and xylem like this. Wavy cambium is formed. Fine. So pericycle will give rise to lateral roots and cambium. In dicot root, in monocot root, this will give rise only to the lateral roots, not to the cambium. Clear, right? Yeah. Any doubts anywhere, ma? No. Now, dicot and monocot stem we will see. So here you see first dicot stem. Dicot stem. See, epidermis will be having multicellular epidermal appendages, unicellular or multicellular epidermal appendages. Those are called as what? Trichomes. Fine. And here epidermis is covered by what? Thick cuticle. Epidermis is covered by a thick cuticle. This cuticle is impervious for both gaseous exchange and water exchange. Next to the epidermis, we have what? Hypodermis, which is collenchymatous. Hypodermis, which is collenchymatous. fine. And then you have general cortex, which is parenchymatous. After that, endodermis is having starch sheath. Okay, endodermis is having what? Starch sheath, which stores food material. Endodermis has starch sheath which stores food material. Next to that, pericycle. Pericycle has semi-lunar projections. Pericycle is made up of what? Sclerenchymatous layer. Pericycle is sclerenchymatous. It has semi-lunar patches which is made up of sclerenchyma. Fine. And then medulla. Medulla, that is parenchymatous. Vascular bundles here, if you see how vascular bundles are arranged. Primary xylem we will observe first. Metaxylem is towards the periphery, that is pericycle. Protoxylem is towards the pith. Okay. Protoxylem is towards the center or pith. Metaxylem is towards the periphery, like this. Protoxylem is towards the center, metaxylem is towards the periphery. So this pattern is called as endark. Endark xylem is present. And how vascular bundles are arranged? Conjoint, just now we have seen, right? Conjoint, collateral and open type of vascular bundles are observed. Fine. Medulla and medullary rays are parenchymatous. Take down. Medulla and medullary rays are parenchymatous. Clear, right? Any doubt anywhere, ma? Yeah. Done. And next we will look into the monocot stem. See, highly complex tissue arrangement you will see only in dicot stem. You have to remember the type of tissue which is present in each and every region. Okay. Next is what? Monocot stem. It is very simple, just see. Yeah, here is the monocot stem. Just look into the dicot stem. Monocot stem, here you see, ma. Epidermis, as it is, it will be having cuticle layer over it, whereas hypodermis here, unlike dicot stem where it is composed of collenchyma, here hypodermis and monocot stem is composed of what? Sclerenchyma. Hypodermis is composed of sclerenchyma. Sclerenchymatous hypodermis. Okay. And then here vascular bundles are schizolysigenous in origin. There is no endodermis, no pericycle, there is no medulla or medullary rays. Fine, right? Vascular bundles, how they are arised, ma? Schizolysigenous in origin. Schizolysigenous in origin. What does it mean? That these parenchymatous cells which are in the general cortex, few of them will be lysed or degenerate, which will create some cavities or spaces from which vascular bundles are originated. What type of vascular bundles you will observe in monocot stem? Vascular bundles are conjoint and closed. Vascular bundles are conjoint and closed. Clear, right? Any doubt anywhere? No. Next. Lastly, we will have the comparative analysis between dicot and monocot leaf. First, we will see the dicot leaf. Okay. So, look at here. Dicot leaf will be having dicot leaf. It has two surfaces of epidermis. The upper surface is called as adaxial surface. Okay, upper surface is called as adaxial surface or dorsal surface. Lower surface is called as abaxial surface or ventral surface. Okay, so the upper surface is what, ma? Adaxial surface or upper surface or adaxial surface. Okay, adaxial upper, or this is also called as dorsal surface. Fine. Abaxial epidermis, lower surface, or ventral surface, or abaxial epidermis. Fine. And here there is of course in the epidermis we have epidermal appendages. Epidermal appendages are not over here. Epidermis is coated by cuticle. Fine. And in epidermis you will see the epidermal structures called as stomatal apparatus over here. In epidermis what are present, ma? Stomatal apparatus. Stomatal apparatus includes stomatal pore, fine, and guard cells and subsidiary cells. Fine. They have a stomatal pore, guard cells and subsidiary cells. So, guard cells in the dicot leaf are, they contain chloroplast and they are what? Bean shaped. Guard cells are bean shaped and they contain chloroplast. They have two wall layers. Outer wall is thin and extensible, whereas inner wall of the guard cell is thick and inextensible. Subsidiary cells are specialized cells which are surrounding the guard cells. So, these three together, stomatal pore, guard cells and subsidiary cells will form up what? Stomatal apparatus. So, this stomatal apparatus or stomatal distribution is more towards the abaxial epidermis and less in the adaxial epidermis. This is an adaptation to prevent the process of transpiration. Okay. So, more stomata you can see in the abaxial epidermis. More stomata is observed in the abaxial epidermis. This is an adaptation to prevent the process of transpiration. Fine, right? And then now looking into the cortical region or the ground tissue system of leaf, which includes what, ma? Purely mesophyll cells. So, mesophyll cells are differentiated here into what? Palisade parenchyma and spongy parenchyma. Mesophyll cells are differentiated into what? Palisade parenchyma and spongy parenchyma. Clear, right? Yes. Palisade parenchyma are below the adaxial surface of the epidermis, which are vertically arranged and there are no intercellular spaces in between them, whereas spongy mesophyll cells or spongy parenchyma are close to the abaxial epidermis, which are arranged below the palisade parenchyma and they have large air cavities in between them. Okay. So, this is about ground tissue system or fundamental tissue system in the leaf. And then vascular tissue system includes vascular bundles. Vascular bundles here xylem and phloem, which are conjoint and closed type of vascular bundles you can see. conjoined and closed. Fine. Surrounding the vascular bundles, you can observe a single layer of bundle sheath cells here. Okay. Surrounding the vascular bundles, you can observe a single layer of the bundle sheath cells in the dicot leaf. Whereas in a monocot leaf, you can observe multi-layered bundle sheath cells. Okay. Monocot leaf will have multi-layered bundle sheath cells, whereas dicot leaf has just single layered bundle sheath cells. Clear till here? So, tomorrow we will look into the monocot leaf, clear differentiation between dicot and monocot leaf, and then we will also finish off the secondary growth from internal structure of the plants. Fine. And after this we will move on to the morphology. Clear, right ma? So, that is all for today's class, and thank you.